Hello, everyone. Welcome to today's live broadcast, Cellular Applications for Image-Based Cytometry on a Multimode Plate Reader. I am Judy O'Rourke of Labroots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by Labroots and sponsored by PerkinElmer. PerkinElmer is a global leader focused on improving the health and safety of people and the environment. Their innovative detection, imaging, informatics, and service capabilities combined with deep market knowledge and expertise, help customers gain greater insights into their science. Before we begin, there are a few instructions. We want to hear from you during this interactive broadcast, so please ask questions or leave us a comment. You can do this by clicking the green Q&A button at the lower left of the presentation window and typing in your comments and questions. We'll try to get to as many as we can, and we'll follow up if we don't have time today. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you can't hear or see this presentation properly, let us know by clicking on the support button on the top right or use the Q&A button. We'll make sure we try to resolve any issues. I would now like to introduce today's presenters, Frauke Haenel and Maria Kuzikov. Dr. Frauke Haenel studied biology and achieved her PhD in 2011 at the University of Kassel, Germany. She joined PerkinElmer in May 2011 as part of the application support team based in Hamburg, Germany. The team provides support for the multimode detection plate readers to customers, sales, and service. Within this team, Frauke mainly focuses on providing assistance on imaging and label-free applications, including training and application development. Maria Kuzikov studied molecular life science at the University of Lübeck. She worked on the development of inhibitors targeting virus replication at the Department of Biochemistry of University of Lübeck and on assay development for studying protein-protein interactions at the European Screening Port. After obtaining her Master of Science degree in 2013, Maria continued to work at European Screening Port, now Fraunhofer IME Screening Port, and was responsible for development of cell-based and biochemical assays in high-throughput screening, HTS, formats, as well as high-content assays in early ADME tox profile studies. I will now hand over to Dr. Haenel for her presentation.
Thank you, Judy, for the nice introduction. During my talk, I will focus on cellular applications for image-based cytometry, and I would like to highlight 5 reasons why you need imaging on a plate reader. And for that, let me first start. So what is cytometry actually? So the International Society for Advancement of Cytometry defined it as a quantitative analysis of cells and cell systems. This means it measures the optical properties of cells, which are the cell size, the cell morphology, the cell count, the cell cycle phase, DNA content, or the existence or absence of specific proteins on the cell surface or in the cytoplasm. The cytometry uses various detection modes, such as fluorescence or indicator dyes or stainless light scatter. And there are several devices available, for example, flow cytometers, cell sorters, or image cytometers. Probably the oldest form of image cytometer is the hemocytometer, which most of you probably know. What can you do with image-based cytometry? So common applications are you can assure assay quality, you can measure proliferation or cell migration, you can detect a viral infection, you can run cell health and toxicity assays. You can analyze transfection or analyze spheroids or other 3D cell cultures and small animals and many more. I will highlight some of them later on. So if we talk about image-based cytometry, we actually talk about it in terms of that we have this technology on a plate reader. So, for example, on our Insight multimode plate reader, it contains of conventional technologies such as absorbance, fluorescence intensity, luminescence, time-resolved fluorescence, alpha technology, and label-free. And on top of that, it has now image-based cytometry. And this is actually, so to say, a bridge between our high-content screening system and the multimode detection world. Our system uses various detection modes for imaging, which is fluorescence, imaging, bright field imaging, and digital phase contrast imaging. It generates one or more images per well of a 96 or 384 well microplate, and it analyzes images automatically during measurement. But now you may ask yourself, why do I need imaging on a multimode plate reader? And I would like now to go through 5 reasons and show you the advantages of having imaging on a plate reader. And the first one will be seeing is believing, so where you have quality control on your plate reader. A second reason is you can take new directions, meaning you can run orthogonal approaches. The third reason is it is very fast imaging technology. You don't have to do it yourself, so meaning I will show you the advantages of automated versus manual imaging and analysis. And another reason is that you can do more for less by using your resources efficiently. So let's now have a detailed look at the first reason, seeing is believing. And it is indeed cell biologists, researchers that work with cells are actually quite lucky because they can visually inspect their model system. So you can always have a look at your cells, what they are doing, how do they look, do they feel good or bad. And by doing that, if you have imaging on your plate reader, you can always check the cell conditions, so meaning you can pre-screen your samples for cell seeding and cell handling errors. You can verify the health status of cells during experimental steps. And as you can see there in the images, for example, on the left side, the image shows healthy cell layer, which results in a confluency of approximately 32%. So there the confluency was determined automatically by the imaging system. In contrast to that, in the middle, there was a bubble trapped in the well during seeding, and you can see that there the confluency was reduced. Or another example on the right, you can see that the cell layer was stretched, and there you can also see that the confluency goes down. So by having these comparisons between wells and confluency, you can uncover artifacts. Of course, you can also use this readout to reduce sample preparation efforts. So you can determine cell number directly in the assay plate, and if you run any other cell-based assay, and you can normalize conventional cell-based assays with this imaging data. Another example is that imaging can detect artifacts such as bacterial contamination or also aggregating compounds. And what the imaging does, or the analysis does, is actually it uses a texture parameter in the image which can detect if, for example, bacteria are present or if compounds were aggregated. What you can see on the left is there you see MCF7 cells which are contaminated with bacteria, which you actually see just as a grayish shade. But the roughness range factor on the top, you can see it's much lower than on the right for healthy cells. So by using this, we are able to determine or to detect artifacts such as contamination or aggregating compounds or anything else that has an influence on the texture in the image. Another very common issue I often observe is that we get images from customers and they tell us, well, I expect a transfection efficiency of 80%. I'm pretty sure it is 80%, but then if you look at cells, you often see that it is not 80%. It's way lower. So there is a good reason to image cells just right before your actual cell-based assay to make sure what amount of cells are really successfully transfected. But I won't go into detail here because Maria will tell you a lot more about this later on. Let's now switch to the second reason. So you can, of course, take new directions and use also orthogonal approaches. What we have done here is we have used several parameters or several imaging modes and also in a conventional mode to look for cytotoxic effects on cells. So first of all, we have we looked at the cell morphology using stainless Brightfield imaging. We used nucleus characteristics by staining the cells in the nuclei and looking at the fluorescence. We used the cytoplasmic enzyme that tells us something about its activity if it is fluorescent, and we also looked at the ATP levels of cells. And what we did in detail is, so we seeded HeLa cells on a plate, incubated them overnight, and after attachment of the cells, we imaged them in Brightfield mode and analyzed the cells. Then we added DMSO, which was our model component or compound, incubated the cells again, and imaged them on the next day in Brightfield mode again. After that, we added the stain for the nuclei and the cytoplasm and imaged the stained cells in fluorescent imaging mode. And on top of that, we removed part of the supernatant, added ATP light kit solution to determine the ATP levels in these cells via luminescence. And here you can see some example images of the Brightfield confluency analysis. And what you see on the left, these are untreated cells after 24-hour incubation. There the confluency is pretty good at 65%. In contrast, treated cells with DMSO have a confluency of 29%, which is mainly due to a rounding or roundup of the cells. So here you see that you can easily apply the analysis automatically, and it discriminates between empty image areas and areas covered by cells automatically. The fluorescent imaging on these cells looked like these images here. So on the upper row, you see the untreated cells, either stained with Höchst or calcein, and an overlay of both. And the lower row are the DMSO treated cells. And what you see that the untreated cells have a very homogeneous nucleus and cytoplasm staining, and the treated cells show a very intense stain and small nuclei, and only a few cells are positive for the calcein stain. And these cells were examined using a preset analysis method on Kaleido, which is the software of the Insight plate reader. And what it does in detail is what you can see on the left in the first image is that first the nuclei are detected via the nuclear stain Höchst, which is indicated here by a white ring around the nucleus, and then a second ring structure is actually produced around the nucleus, and this ring structure analyzes the intensity of the cytoplasm stain, which is in our case the calcium AM. And if you apply this analysis to treated and non-treated cells, you get an idea what's happening with the nuclei and the cytoplasm stain. And in the end, we ended up with this table of EC50 values. So we evaluated the cells with dose response curves, and there you see that in some cases we have very similar EC50 values, but in other cases, for example, for the cytoplasmic stain, the calcium there, the EC50 values are much lower. And what you can clearly see is here that depending on what parameter you use or at what detail of apoptosis, for example, you look at, you see that there are differences. And in many cases, it is known that, for example, if you would run an ATP analysis only, it can happen that compounds interfere with the biotechnology and with the biochemistry, and in this case, it can make sense to also look at these cells with imaging. Another reason why image-based cytometry is very helpful on a plate reader is actually because it takes almost no time and is very, very fast. And this is mainly based on the short measurement times that that the imaging technology has. So as you can see on the right, for 96 well plate we need less than 2 minutes, and for 384 well plate we need less than 5 minutes. And short measurement times are combined with automated image analysis during measurement, and because we work in high density microplates, this is also an advantage in terms of saving time and money. So yeah, the combination of these points leads to a minimization of disturbances during measurement of the cells. And probably many of you remember long microscope sessions where you actually had to examine one sample after the other or well by well. You can amplify these time savings in applications which involve the analysis of multiple time points, so if you run kinetics. And you can quickly test multiple assay conditions in parallel, but this is also something Maria will talk about in more detail. A nice example of fast imaging is also working with small images, with small animals, because these are often still analyzed manually. And what you can see here is an example image of 2 transgenic zebrafish embryos in a 96 well plate. And what you can see, so we applied Brightfield and GFP fluorescence imaging. And what you can see, one of these embryos is successfully transfected with GFP, and the other is not. And we used here a custom image analysis method, and this method revealed that the object coverage is 11%, which is corresponding to 2 embryos here, and the fraction of the fluorescent area is 57%, so which means that one of the embryos is positive for GFP. So the benefit of the analysis here is you can verify the GFP expression automatically in zebrafishes, and you can quantify the object per well. Another advantage of the fast imaging is also that you can easily run image-based proliferation assays. So what you can see here are fibroblasts and smooth muscle cells, which were not treated at all or treated with a proliferation suppressor. And we periodically determined the confluency over 50 hours here. And this can be easily applied to any cell-based assay, so that you either you use it as a main assay result because you want to measure the proliferation of cells, but you can of course also use these results as a starting point, so to say, for another cell assay. So, for example, if you say, okay, I will have to wait until my confluency reaches 90%, and then you can track that with the imaging module and can start your assay. And here it's really a nice advantage to be able to determine the confluency and number of cells with the Brightfield mode or a digital contrast mode, where you don't stress your cells at all. The fourth reason for image-based cytometry on a plate reader is that you don't have to do it yourself, so meaning there is automated image analysis available. And this one here, the viral infection or the Plaque assay, is a nice example from a customer. And there what you see on the left are actually infected cells, and whenever a cell is infected, the virus produces a plaque in the cell layer, and the plaque can be seen here on the left in green. So the viruses are stained with an antibody, and the fluorescent plaque represents a number of infectious particles, for example. And using the automated analysis, you can count these plaques automatically. And in the past, these customers, they were not able to do that automatically, and there several people were counting these plaques manually, and which and you can see here that it is quite a huge advantage in terms of the workflow that is used here. Another assay that we have run together with the Fraunhofer institute is a fully automated Oris cell migration assay. So this Oris cell migration assay is an assay where you first put some stoppers into each well of the microplate, then you seed the cells and let them settle down, and the cells are not allowed to attach to the area of the stopper. And when you want to start your migration assay, then you remove the stopper and a hole in the cell layer is left. And this hole can be analyzed automatically via imaging. You see that on the cell picture there. And what it does, it automatically detects the cell-free area in the center and then determines the confluency in the center area automatically. And by doing this, you can then determine cell migration of several cells, or in terms, or in when you have your cells treated with different compounds. And what you see there in the dose response is that you can, or a result of this analysis is, for example, the gap closure velocity, which can be used as an indicator for migration. In our case, we have really fully automated this assay. So we used a plate handler workstation, which transferred the plate every 2 hours from an incubator to the instrument. Let's now come to the last reason, which is that you can, with image-based cytometry, you can do more for less by using your resources efficiently. And what I mean by that is, for example, that you can free your high-content system for more advanced work. So whenever you have high-content images, of course, biological relevance increases, the content increases, but also the cost and the time. And by having image-based cytometry on a plate reader, you can use this plate reader actually to run readouts at a lower level, and then move these samples over to your high-content imager. Usually, if we run 3D cell cultures, we use the Spherold 3D culture system. And this results really in very, very nicely shaped spheroids, as you can see there on the images from the inside. And these spheroids were grown in a hanging drop, which is the spherogravity plus plate. And then they are transferred in the gravity trap plate. And this is what you see. So there you see a fluorescently labeled spheroid in a gravity trap plate. By the way, so these are spheroids that were grown directly in the gravity trap plate, so not in a hanging drop. So there you can clearly see the shape is not as nice as on the images before. But still, we use these spheroids to treat them with FCCP, which is an inhibitor of the mitochondrial oxidative phosphorylation. And you can see in the images the FCCP concentration increased from left to right. And the upper slide shows the images of the inside, which has a 4x objective. And in contrast to that, the lower images are from the Operetta CLS, which uses in this case, used a 10x lens in confocal mode. These spheroids were stained with TMRM, which is a fluorescent marker for the mitochondrial potential. And you can see with increasing FCCP concentration, this marker intensity decreases. And if you then compare the evaluation of these images of the two instruments, you can see that they nicely correlate. So we have almost the same EC50 value for both these instruments, with the same samples, with a good fit. So here between Operetta and Assays, we have very, very similar results for wide field and confocal analysis. And this clearly shows that you can actually use image-based cytometry for early and low-level analysis, but you would then hand these spheroids, in our example, over to the Operetta to do more advanced work, to have a look at the cells in the spheroid in detail and so on. And with this, I hope that I or that you now have a better understanding of the value that you can get having image-based cytometry on a plate reader. And if you want to recap the information, so we have soon, or we will have soon, a white paper available on our Insight microsite. So you see the link there. If you are interested in that, please feel free to look for this white paper there. And I would now like to hand over shortly to Judy. And thanks for your attention so far.
Thank you, Dr. Haenel. I would now like to hand over to our second speaker, Maria Kuzikov.
Thank you. Well, in the second part of our webinar today, I would like to show you an approach for optimization of a transient transfection protocol using Insight. And well, before we come to the main part, I'd like to give you a short introduction about our site here in Hamburg. Then we will talk about challenges and goals of the transient transfection. After that, we will come to the final part and have a look at the optimization of a Brightfield transfection protocol on the Insight. And well, last but not least, I will try to sum up all the lessons learned topics. Well, since 2014, we are here part of the big Fraunhofer Society, so actually part of the Institute for Molecular Biology and Applied Ecology, which has 6 sites here in Germany and 2 international ones. And well, our main goal is actually to bring the academic research and the industry more closer together. And our main field is the screening and assay development. Well, as a huge part of our screening is based on cellular assays, we of course have a lot of challenges, as well as a lot of experience with challenges and goals of transient transfection. Well, when we are speaking of transfection, what does actually mean by that? So we try to introduce a foreign gene in a cellular system and try to study its regulation or also the gene product. And in fact, we divide it in 2 main classes. So one is the stable transfection, where we integrate the gene in a host cell, and then the gene remains after the replication, or we can use a transient transfection. There, the gene is only in the cell for several times, so for several periods, and it's not integrated in the genome. You can use various methods. So, for example, the biological by induction of the gene of interest via a virus, or classical chemical approaches with cationic lipids, or like physical, the electroporation. But always the main goal is to have a high expression of the gene of interest with low cytotoxicity or minimal effects on the cell, so especially when you're using primary cell lines. And of course, there are more things to think about when you use transient transfection. So besides the cell line, or whether it should be a transient or a stable transfection, you have a lot of possibilities to optimize it. So first of all, of course, the transfection conditions. So which transfection media you can use, so the cell number or the transfection reagent. On the other hand, also the ratio of the plasmid to transfection reagent, or when you're using different plasmids, so like more than one, then of course the ratio from plasmid to plasmid. Another point is, of course, the incubation time of the transfection complex, so the transfection reagent with the plasmid, or also with your cells. And well, maybe also whether you want to use a batch transfection or you transfect already plated cells. So for example, in our case, it's always easier to use the batch transfection. And besides the main goal that you have a high expression level of the gene of interest, it might be also necessary to optimize other options. So for example, in our case, we normally use the transient transfected cells for the text of compounds. And there we have, of course, to pay attention that we have a low plate-to-plate variability or day-to-day variability in our assay. Then we have a good quality of the assay. So, for example, that the Z prime value is high enough. And, of course, the issue with the cost and time. So we made a good experience with the reduction of time by tracking multiple quality controls parameter. And actually, in the last month, we used the Brightfield transaction analysis of the N-site to do it. And what you see now on the next slide is the general transaction protocol. So it's normally a 3-day event. On the first day, we harvest our cells, prepare the transaction mixture by using different plasmid concentration transaction reagents. Then we mix it together, and then we plate out the cells. So that's our batch transaction approach. And on the next 2 days, we then quantify the transaction efficiency by using the Brightfield transaction analysis on the N-site. So what you have to do for it is, of course, first of all, to optimize your exposure parameters. In the left upper corner, you see that you can choose different channels. So the first one is normally the Brightfield for normal cell detection. The second one is depending on your tag of your protein. So in our case, it was a GFP tag, so that's why we used the blue light source. In the middle, you see our exposure parameters. Well, of course, you may have to optimize it for each single experiment, but these were the parameters we used for our analysis. In the right upper corner, you see then the plate layouts, where you, of course, select the wells you want to image, and then below it, you see 2 buttons for selecting the channels, whether you can so you can use the overlay or have a look at the channels separately. So we made the experiment that it always is good to have a separate view on the channels, as you may have difficulties to see the signal in the overlay. Then in the next part, you have the possibility to run an automated analysis, which was mentioned by Frauke. So first of all, you select the well or the region of interest. So by selecting the 3 options on the bottom. And in the next part, you can then detect the cell covered region by choosing the threshold parameters. Then you actually do the same for selecting your transaction region. And here it's always good to have a negative control, so the non-transfected cells, then you can see easily where are your really positive transfected cells. At the end, you will get a table which gives you the percentage of transfected cells, of course, but also parameters like percent confluency, the mean signal, or also texture properties. For example, the roughness parameter may give you a hint about how toxic your conditions are. So, of course, as I already mentioned, there are a lot of things which may influence your transaction efficiency. We made good experience by using a so-called transaction matrix to test as many conditions as possible. So here in this case, we have 3 different cell lines. So the HEC cells have G2 and QK1. Tested at 3 different cell numbers with 2 different transaction reagents and 3 different plasmid concentrations. So you see you can run really multiple parameters at the same time. So first of all, we normally have a look at how healthy are our cells after the transaction, because normally we use serum-reduced conditions for the transient transaction, and it may already influence your results. So for example, you see that HEC cells are a bit rounded up. So they are normally more sensitive to changes in transaction conditions or like cultivation conditions. In the same time, you can have a look on the cell number. So for example, when you have a look at the HepG2 cells and the HOK1 cells, you see that too high cell number may result in clumps formation or, like, let's say, colonies formation, which may also influence your transaction efficiency. In the next step, it's also good to have a look at the influence of the transaction reagent itself. So also in this case, you see that the HEC cells, for example, are more sensitive to the transaction reagent 2, and it results in the reduction of the confluency, and also the cells look more rounded up. And in contrast to it, the HepG2 cells, well, are not really changed in their morphology. When we have a closer look, it's of course easy then to see clear numbers, as always to just have optically analysis of the images. You see the results for all 3 cell lines, where the clear colored bars are representing the confluency and the spotted ones, the roughness parameter. As previously mentioned, the roughness parameter gives you a hint also about the toxicity. So if it goes up, then we have an increased number of rounded up cells. And you can easily see that the HEC cells are, as expected, more sensitive to the increased plasmid concentration. So the confluency goes down and the roughness value goes up, whereby HepG2 cells and CHO cells show only, well, low changes in those values. In addition, you also see that lower cell number seems to be more sensitive to changes in cultivation or transfection conditions compared to higher cell numbers. Well, of course, it's good to see that we can track the confluency parameters and so have a look at the cytotoxicity of our transaction parameters, but the main factor is, of course, the transaction efficiency itself. So when we have here a closer look, then, for example, you see the application of 2 different transaction reagents in HEC cells and the HOK1 cells under actually the same transaction conditions. And here you see that just the change of transaction reagent can result in really high increase in transaction efficiency. So in HEC cells, compared to CHO cells, they just show no difference at all. Well, on the next slide, we'll have a look at the real results. So here you see again our 3 cell lines, and now the clear bars are representing the transaction efficiency, and the spotted one the confluency, again for the tracking of cytotoxicity. Also here you can clearly see that HEC cells gives us the best cellular system for the transaction, so we can reach up to 80% transaction efficiency when we use 6 nanogram per microliter of our plasmid, whereas the HepG2 cells and CHO cells show only limited transaction efficiency under 10%. At the same time, you see that when we use 2 nanogram per microliter plasmid, we have the best results in all 3 cell numbers in HEC cells. So with other words, that we would have to optimize the transaction conditions for HepG2 and CHO cells, but with HEC cells, these are quite good results for the moment. Well, as I previously told you, that the change in transaction reagent can result really in strong increase in transaction efficiency. And here it's, well, one more the proof of the concept. So on the top you see the transaction reagent 1, and below the results for transaction reagent 2. You can clearly see that with transaction reagent 2 we have really good transaction efficiency results. Also we have a bit decrease in the confluency, but at the same time there might be a hint that with transaction reagent 1 and the lowest concentration of the plasmid, we also might have good transaction efficiency results. But when we have a look at the images, which gives us an additional quality control, you see them on the bottom of the slide. You can clearly see that the conditions for transfection reagent 2 results in enormous clump formation, and that's why giving us this quantitative better transfection efficiency result. So you see that it's always good to have an additional quality control by imaging your well. Well, the first result, I hope you could see that you can use the Brightfield transfection analysis to track in parallel the transfection efficiency and the cell viability. In the same time, you can use the transfected cells for different approaches. So you can also run additional assays in parallel as the imaging is not invasive, and you can use your optimized results for an additional analysis. So, as we saw previously, the hex cell line was the best cellular system in this case, and of course, it is good that we can run multiple parameters in parallel, but it's also good to have an additional experiment to prove the result or to confirm the result. And here on the next slide, you see a repeat of the previous experiment. So we use the hex cells with the same conditions, so with the same cell numbers and plasmid concentrations, and we could prove that the obtained results from previous experiment give us nearly the same transfection efficiency. So that's why in our case it was good to prove that the plate-to-plate and day-to-day variability is really low. In the same moment, we wanted to see whether we can increase the transfection efficiency by increase of the transfection time. And indeed, when we have a look at the three different cell numbers, we see that by the induction of transfection time, but only 24 hours, we have an increase of 20% of the transfection efficiency for all three cell numbers. Actually, also achieving almost 100%, so 90 to 100% when we use 20,000 cells per well and 2 nanogram per microliter plasmid. And at the same time, we saw that the confluency is not changed, so that the cells are quite happy during this experiment. Yeah, so that's how we optimize the transient transfection in our hex cell line. Well, last but not least, I will just maybe mention one additional possibility how you can track the transfection efficiency. So I often see the problem that we have not tagged proteins, so we cannot easily track them with the imaging. So what we also use is the fluorescence labeled transfection reagent, which increases the fluorescence when it is integrated in the membrane. So normally the signal, the GFP signal, is of course much higher, but still with the NSAID you can easily detect the amount of positively transfected cells in those cases. So all in all, we have now seen that by using the NSAID we can track in parallel the transfection efficiency and the impact of the transfection complex, so see the impact on the cell viability. We also saw that we can use the same batch of transfected cells, and with this non-invasive methods we can also use our transfected cells for downstream applications, so for the real functional readout. Well, with other words, you can really easily reduce the time and the effort you need for the optimization of the transfection protocols. Well, that's it from my side. Thank you for your attention.
Thank you for your excellent presentations, Dr. Frauke Haenel and Maria Kuzikov. Before we get started on the question and answer session, I would like to remind our audience how to submit questions. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. And our first question is, in regard to fluorescent imaging, what colors can the Insight use?
Okay, this is probably a question for me. So the colors that can be used is, so we have, you can have emission in the UV range, in the blue-green range, or in the red range. A big advantage of the Insight is that we use a triple band setup, so which means we don't have to switch filters all the time, which is a result, or which results in a very, very fast measurement time.
If I get so many different parameters and readouts from my experiment, how do I know which of these is the right one?
The advantage is actually, it is to have these different parameters. So it always depends on what you look at, what you want to look at, and you would compare the same parameter with for different cells for different conditions. But you have the full picture of the cell and how they behave under certain conditions.
For the zebrafish analysis, you mentioned that there was a custom image analysis used. What does this mean in detail, and can you provide custom analysis methods for your plate reader?
Yes, that's true. So we can provide custom analysis methods, so which means so the Insight, it provides preset image analysis methods for most of our very common applications. But if customers run exotic applications which cannot be evaluated with the preset ones, then our software team here, our imaging software team, they can provide custom methods as well.
It was mentioned that the Insight needs less than 5 minutes per plate. Is this measurement time dependent on the number of excitation sources? And if yes, how?
Yes, it's always dependent on the excitation sources, because if you, for example, have a very low or very dim stain, then you will need a higher exposure times, so which can increase the measurement time. And of course, if you also increase the channels that are run, then then if you increase that number, then the measurement time can also increase.
What do the nucleus area and the nucleus intensity tell you about the cells?
Sorry, can you say that again, please?
Yes. What do the nucleus area and the nucleus intensity tell you about the cells?
Maria?
Yeah, maybe I can answer this question. So the nucleus area gives you a hint about the cytotoxicity. So normally when you see apoptotic events, then you have the shrinking of your cells that you will also see in the nucleus. Here you have to pay attention that also the DNA density increases, so that's why it might be that you see a higher intensity of your dye in the nucleus. So it's like an additional parameter for cytotoxicity.
How does the image analysis work in detail, and is it easy to set up?
So the image analysis in our hands works pretty straightforward. So of course, it's always good that when you have positive and negative controls, so like wells where you clearly know what you expect to see. So for example, in our case, non-transfected cells. And actually, you can also run the image acquisition and then change your analysis afterwards. So you have always the possibility to go back and then recalculate the assay, so in case you are not sure.
Can you also do label-free imaging?
I'm not sure if I understand the question correctly. I assume our label-free technology is meant. So, and what you can do there, of course, you can combine the label-free measurement with imaging. So the label-free plates can easily be imaged on the inside as well, and you can combine it, which is in many cases very, very helpful to understand or to track the conditions of the cells in the label-free plate. So their combination is very helpful.
So your insight is like an incubator, or you need to put it in an incubator?
So the instrument can be set to 37 degrees. So you can increase the temperature inside the instrument, which you can use for long-term experiments, for example. But as we have shown, you can of course also automate it. So automate it and put it back into the incubator. And many customers also, for example, track their cells regularly and put them in the incubator for a few hours and put the plates back in between on the inside. And as the measurement times are so fast, that's not an issue with the cell condition.
Do you have a software to count automatically?
Yes. The software counts automatically. Yeah.
Is it possible to detect cell numbers without nuclei staining?
Well, I think what you count will be the percentage of confluency. So it's linked to the cell number, but it's not the direct calculation of the cell number you might want to use.
Does the Insight image the whole well in one field of view, or is it scanned with multiple fields of view?
It has a 4x objective, and it scans the whole well in one view. So that's that's very fast. So you don't have to set up several fields or several fields of a well. So you really have one shot per well, which makes it pretty fast.
It appears that the Insight system lends itself only to image analysis of adherent cells. Is there any way to use it for cells in suspension?
Yes, cells in suspension is also possible, of course, and you just let your cells settle on the well bottom, the suspension cells, and you can image them as well very nicely and count them. And
I'm sorry, I did not mean to interrupt you there. Please.
Sorry, no, that's fine.
I will just pause for a moment and see about additional questions coming in. I will say that, let's see. While we're waiting to see if additional questions come in, I would like to once again thank Dr. Frauke Haenel and Maria Kuzikov for their presentations. Do you have any final comments?
No, not from our side.
Actually, not really comment, but we would like to thank you, the audience. So I saw we had quite a lot of people attending, so it was really nice to have audio.
Yeah, thanks for listening and watching.
And of course for moderating.
Yeah, thank you, Judy.
Thank you. Thank you both. I would also like to thank our sponsor, PerkinElmer, for making it possible to bring this presentation to you. Today's webcast will be available for on-demand viewing through April 2017. You'll receive an email from us alerting you when it's available on demand and posted on labroots.com. You are welcome to forward this announcement to any colleagues who weren't able to join in today. Thanks for logging on and participating in today's broadcast. See you next time.